I have now to switch in English, and I am now extremely pleased to introduce our guest speaker, Professor Michael Hall. Michael Hall is born in Puerto Rico and spent his childhood years in South America. He received his PhD from Harvard University in 1981 and was a postdoc fellow at Pasteur Institute in Paris at the University of California. He joined the Biozentrum of Universität Basel in Switzerland in 1987, where he is currently professor and former chair of biochemistry. Since 2023, he is also a distinguished principal investigator at the Institute of Human Biology of Hoffmann La Roche in Basel. In 1991, Michael and his colleagues discovered the mechanistic target of rapamycin, mTOR, has subsequently elucidated its role as a central controller of cell growth and metabolism. mTOR plays a very important role, a key role in development and aging, and is implicated in various disorders, including cancer, allograft rejection, and diabetes. Mike Hall is a member of the United States National Academy of Science and has received numerous awards. Here, just a very brief summary of this long list, including the Louis Jeantet Prize for Medicine, the Breakthrough Prize in Life Science, the Canadian Gardner Award, the Albert Lasker Award, the Siebert Prize, and last but not least, last year, the Balzan Prize. Mike, it's a great honor for us. Please, on the stage.
Sale sul palco Michael Hall, professore di biochimica presso il Biozentrum, Università di Basilea, Svizzera. On stage, Michael Hall, professor in biochemistry at Biozentrum, University of Basel, Switzerland.
So, distinguished guests, dignitaries of Humanitas University, friends, colleagues, ladies and gentlemen. An immense pleasure and honor to be here today to celebrate the opening of the academic year of Humanitas University. And I thank Professor Terracciano for the invitation, wherever you went to. Okay. So, today, I would like to tell you a personal story. It's a story of discovery. And what is this story about? You'll see, and as Professor Terracciano briefly described, it's a discovery of a protein that plays a very important and fundamental role in the cell. And this protein is called TOR, and I'll give you more details on this soon. This very important fundamental role it plays in the cell is to control the growth of the cell, and this is both in health and disease. Now, I'd like to present this story more than just a story of discovery, but also as a story of the importance of fundamental curiosity-driven research. Indeed, I'd like to present this story an example of much of what you've already heard from the speakers that came before me, that fundamental research is absolutely critical. So in a sense, I'm simply here to confirm everything that you've already heard from what I consider the very enlightened visions of your leaders, your ministers, your politicians, your governors, your mayors. But in addition to this being an example of fundamental research and the importance of fundamental research, what I have to say has a third element, and that it is also, I hope you will find, is a story of success. Thus I'd like to end the talk, after you've heard, reflecting on what are the elements that make success. And this has already been brought up earlier today. And again, I think I'm confirming what you already heard. I will talk about what led to success in the story I will tell you, and perhaps is there a formula for success, and can that formula be transplanted or transferred to other environments? Can this be of help to others? But I'd like to start the story by telling you about cells, and the particular cells I'd like to tell you about are biological cells. And the point I would like to make is that cells are the unit of life. So they are the smallest and simplest life form on the planet. And this point is made by the fact that in some cases, organisms are nothing more than a single cell. And the example of this is a yeast cell, the same yeast we use to make bread. This is the whole organism. It's nothing more than one cell. And of course, this is a living organism, so it tells us that a single cell is the unit of life. But in many cases, there is more than a single cell in an organism. For example, a mouse has 3 billion cells, many, many more cells. And a human has 10 trillion cells. This, of course, is a very large number. Most people don't even think about numbers on this scale. But in every case, despite the fact that you might have many, many cells to make you as an organism, every one of the cells of these 10 trillion or 3 billion is also a unit of life in itself. And this is proven by the fact that you can take a cell out of a body, a human body or a mouse, and you do this in the laboratory, and you can culture that cell in the laboratory, and that cell will live by itself independently of being in the environment of a whole organism. Again, demonstrating that a cell is a unit of life. Now, however, this creates a new problem. And that is, if every cell is a unit of life, how do you coordinate all these cells in an organism where you have, let's say, 10 trillion of them? How do you coordinate their growth, their division, their replication, their propagation, such that they know to generate an organism with a proper number of cells, and not only that, but every one of those cells has to be the right size, such that humans are more or less very similar to each other, and not only that, but the organs within each one of us are a certain size which have the right proportion to every other organ in our bodies. So this is an immense problem. We know how difficult it is, for example, to govern a city like Milano. Imagine governing a community of 10 trillion entities. This is a mind-boggling task. So before I get into the cell further, I'd like to actually show you what a real cell looks like. So this is 2 cells. Here's one cell, here's another cell. And these 2 cells are about, what you see here are 1 million times bigger than a real cell. So real cells are much smaller than this. And this cell has, like all cells, these 2 cells have a nucleus. So this is a nucleus here, and this is what contains the chromosomes or genes. And then the green part here is the rest of the cell, and this contains what we call the cytoplasm, and this is where most of the, I would say, biochemical reactions that occur in a cell happen in this part of the cell. And surrounding the cell, there's a membrane, which is the eliminating barrier between the cell and its environment. Now, these cells have to, of course, if we start from a single cell, which we always do, a fertilized egg, this single cell has to replicate, and here you see these 2 cells replicating. In other words, they're dividing to give rise to, in this case down here, we see this one cell is divided into 2 cells. Again, there are 2 nuclei and 2 cytoplasms where there was 1. and this is the case for both cells. So if we go into this more schematically, what I just showed you was this process up here. This one cell divided 2 cells, and eventually that would give to 4 cells and so on, until, if you were human, you would reach 10 trillion cells. Now, this is a rather complicated process because it's actually a superimposition of 2 separate, functionally separate processes. And those 2 processes are, this cell has to grow and divide. And the best way to understand that is if we separate these 2 processes, what would happen? So here's a case where the cell is dividing but not growing. And this, of course, would be catastrophic because this would ultimately not be a sustainable situation. These cells would disappear way before reaching 10 trillion cells. You can't divide the mass so many times and still have a viable organism. On the other hand, if you had growth without division, that one cell would get bigger and bigger. And of course, this again is also a catastrophic event. This would be unsustainable. This would not give rise to an organism. So we have 2 separate processes. The one up here captured the interest of researchers already back in the 60s and was worked on very heavily in the 80s and 90s, and was eventually recognized with a Nobel Prize. Understanding how this process is controlled was recognized with a Nobel Prize in 2001, which went to Paul Nurse, Tim Hunt, and Lee Hartwell. Now, this process down here, the growth part, which goes hand in hand with the division part, was largely ignored. For some reason, scientists didn't understand that this is in fact 2 separate processes involving growth and division, because as is human nature, you would focus on one cell, and that cell is always the same size, and you don't see the increase of mass. But in fact, there's obviously an increase of mass here versus here if you look at the whole population. So division part has to be controlled, of course, but the growth part has to be controlled as well. So how are these 2 processes controlled to give rise to an organism of 10 trillion cells? And this then is the controller of this process is what we discovered. And the controller of that process of cell growth is this protein called TOR. And I'll tell you more about it in a moment. But TOR is a protein, which again, this is a cell, which we've opened up a window to look in. Again, the nucleus here, and this is the cytoplasm. This protein exists in several copies in the cytoplasm of the cell, and it receives signals from outside the cell, from receptors on the cell surface, which bind hormones and growth factors or nutrients. And this then transmits this information from the outside of the cell across this membrane through a biochemical cascade, which ultimately turns on the TOR protein. So TOR then can activate all the cellular processes which lead to increase in cell size, cell mass, and cell growth. Now, the pathway via which TOR senses this input from the environment, from outside the cell, is much more complicated than what is shown here. We've been working on this process for actually a little longer than Humanitas has existed, for 35 years. And it's the state of knowledge is more something like this. It's like a very complex electric circuit, not just a single biochemical pathway. This model, I have to say, is already quite old. It's from 2006. It's gotten so much more complex that we can no longer draw it in a single 2-dimensional piece of paper. But also to get back to this issue of what was here 30 years ago. The president told you about there were just nothing but fields here 30 years ago. Well, this intellectually was the same in the TOR field. This was almost 35 years ago. This is the very first ever model of TOR, of this biology of TOR signaling that was proposed. It was proposed shortly after a discovery of TOR in 1991. And it's kind of I would say, a ridiculous model to have been published, because it gives you absolutely no useful information other than the fact that TOR exists. We are very proud of having discovered TOR, although in retrospect we probably shouldn't have published this model. There were more question marks about what it did, what it responded to, and then what it did to ultimately do. We didn't even know what it did in the beginning. It was just a protein that was the target of rapamycin. So this was our empty field, intellectual empty field, 35 years ago. And now we've built this edifice, much like it's been built here. Again, more of an intellectual edifice involving all this increase of knowledge. We've generated an immense amount of knowledge here, not only our group, but many groups around the world now. This has grown into a large field. The number of publications in this field have exponentially increased. Now it's continued to go up. This is an old slide from 2012. Now we're about 7,000 publications per year on the TOR field. So it's become quite a large field, which we cannot even follow all the literature. It's so vast. So TOR, this protein TOR, this name TOR is an acronym for target of rapamycin, as you see down here. So you might ask, what is rapamycin? Rapamycin is this molecule here. It's a very interesting molecule because it's a natural molecule that's produced by a soil bacterium that came from a soil sample that was collected on Easter Island, which is known to the locals as Rapa Nui. And this was a group of scientists from Canada who traveled all the way from Montreal to Easter Island, or Rapa Nui, to collect soil samples because they wanted to isolate some exotic microbe that would make a novel molecule which they could then develop into an antifungal. So they wanted to develop a drug which would treat a fungal infection. They did isolate such a molecule that had antifungal activity. It was this molecule here. But as they were developing it to commercialize it, they found it had an undesirable side effect of suppressing the immune system. And of course, suppressing the immune system is the last thing you want to do to anybody with a fungal infection. So as a result, it was rejected, it was discarded, and largely forgotten as an antifungal. But then about 10 years later, two very brave transplantation surgeons who were developing immunosuppression to make organ transplantation a feasible clinical procedure, were trying to find ways to suppress the immune system so the transplantation patient wouldn't reject the transplanted organ. And they then rediscovered rapamycin in the old literature, which is now already being forgotten from 10 years earlier, and then brought it back, but repurposed for the very reason for which it had originally been rejected, to suppress the immune system. And this was part of the revolution in medicine which led to transplantation surgery. So this very modest molecule that came from the dirt in a very far away place has become very famous. It was first famous as an immunosuppressive drug to treat transplantation patients. It has now become even more famous as an anti-cancer drug. It's commonly used for treatment of breast cancer, renal cell carcinoma. And even more recently, it's been used in treatment of in cardiovascular disease and the treatment of restenosis after angioplasty. So it's rather rare that one molecule can be used in three different major therapeutic areas. So this already suggests that whatever this molecule is doing in the cell, and nobody knew what it was doing, they just knew it had the effect, the desired effect, in the beginning of suppressing the immune system. Nobody understood the molecular mechanism. What does this molecule actually do when it gets into a cell? And that's then when we became interested in this molecule. This is in the late 1980s. We want to understand, what does this seemingly magic molecule do in the cell to elicit a beneficial effect in a therapeutic effect. When we started working on it, it was not yet clinically approved. It was still in preclinical stage of development, but the therapeutic potential was already quite obvious. So how did we isolate the TOR protein? Well, we took cells and we broke them up to all the contents would be released. We made sort of think of making a vegetable soup, but with cells. So you grind up the cells or homogenize the cells. And with the soup, now it's containing a large mixture of all the proteins which are in a cell. There are about 10,000 different proteins in a cell, each one in many thousands of copies. So we wanted to fish out the one protein that rapamycin bound to somehow change the cell to deal with not only treatment of these diseases, but also to have some other effect on the cells, whatever that effect might be. So we, in a sense, we use rapamycin as a bait to fish for the one protein that it bound. We, of course, did not use a fishing rod to do this. We use the most modern molecular techniques. But these techniques allowed us to fish out a protein, which Tor bound with high affinity and specificity. And we call this protein Tor for target of rapamycin. This is a rather neutral name because it does not reflect the function of the protein. It certainly reflects the fact that it is a protein that rapamycin binds. The next challenge was then to understand what is the function of this protein. I'm skipping many years of work, but we discovered that the function of this protein was in fact to control this process of cell growth, which scientists had been ignoring until this point. And they'd largely been ignoring it not only because they did not see the cell growth, but also because those few who did see it did not think there was a regulated process. It was a passively regulated process that happened spontaneously when there were nutrients available. The nutrients, which are building blocks for growth, would go into the cell, and the cell would then use these building blocks to build. An uncontrolled growth process always going on. In retrospect, this is a very naive view on how cell growth occurs because, of course, it has to occur in a very precise and regulated manner. And we know that, of course, today because if it doesn't, then things like cancer happen. And I'll come back to this in a moment. So what we have here is a tissue. And again, you see these individual cells with a nucleus in each cell. And this tissue is the liver of a fruit fly, of a Drosophila. So many of you probably didn't know that flies have livers, but they do. And this experimentalist, Tom Neufeld in the United States, what he did was he selectively inactivated the Tor protein in these two green cells. And you see these cells end up being smaller than their neighboring wild-type cells, their neighboring normal cells. Bruce Edgar, another Drosophila fruit fly geneticist, did the inverse experiment. He now selectively activated Tor again in these green cells. And these are the same tissues. So these cells here and here are the same size. They're just at a different scale here. You can see when we increase Tor activity, the cells get much larger, and we inhibit it, they get much smaller. So the obvious conclusion is that Tor controls cell growth and thereby cell size. Now, Tom Neufeld took this experiment one step further. He isolated a mutant Drosophila, or fruit fly, which now all the cells of the entire fly were defective for Tor signaling. This was not a complete loss of Tor, because if it was a complete loss of Tor, this would have been too severe a defect, and this fly never would have even been born. So it was a partial loss of function of Tor activity. It's a so-called Tor mutant fly. And when Tom Neufeld isolated this fly, he saw that it was a smaller but perfectly proportioned fly. And then he asked himself the question, why is this fly smaller? Is it smaller because it has the normal number of cells, but each cell is slightly smaller? Or is it smaller because each cell is the normal size, but just has fewer cells? So is it due to changes in cell number or due to change in cell size? And how would you ever answer a question like that if you were a scientist? Because, of course, you can't count every one of the billions of cells in an organism and measure every one. So Tom Neufeld, being very clever, he looked in the wing, which is a plane of cells, so you can easily measure the circumference of every cell in an area of the wing, and also count the cells in a patch, a certain area of the wing. And from that, you then extrapolate to the rest of the fly. And what he found was this fly has more or less the normal number of cells, but now each individual cell is smaller. Okay, so the conclusion from this is that TOR not only controls, because it's TOR which is defective, TOR controls not only size of individual cells, but ultimately, as a result, controls size of tissues, organs, and ultimately size of the whole animal. Obviously, a very fundamental aspect of biology. TOR has now been shown to be involved in controlling growth in many other contexts, physiological contexts. One of my favorites is muscle. If you exercise and your muscles get bigger, you don't get more muscle cells, but your existing muscle cells get bigger. And this has been shown to be controlled by TOR. In the central nervous system, there are very dramatic changes not only in cell size, but in cell shape. And this is also all shown to be controlled by TOR. And somebody's even shown that a queen bee is larger than the worker bees in a colony of bees, because the larva which gives rise to the queen bee has actually been fed a super nutritious jelly called royal jelly, which upregulates the TOR pathway in that larvae to give rise to this larger bee. So TOR controls growth very broadly in terms of physiological context, and also very broadly in terms of organism. Essentially any organism in nature, what we call eukaryotic nature, where there's a change in cell size, cell growth, it's probably mediated by a TOR signaling. So how does TOR control growth? Well, I'll get slightly scientific here. It controls a large number of cellular processes which collectively determine mass accumulation and ultimately size of the cell. And these processes can be subdivided into two groups: the anabolic processes, which TOR activates, so protein synthesis, nucleotide synthesis, lipid synthesis, ribosomes. It makes all these things what the cell needs to build, to construct itself. On the other hand, it inhibits all the processes, the catalytic processes, which prevent accumulation of mass. So TOR balances these opposing forces of synthesis and degradation such that the cell accumulates the appropriate amount of mass, i.e. reaches the appropriate size relative to whatever the nutrients might be in the environment. So in very simple terms, TOR is what makes us grow when we eat. Now, TOR also has a rather sinister side to it, because what happens when there's a defect? What happens when, for example, a mutation which might hyperactivate TOR occurs? Well, this leads to many different kinds of diseases, for example, cancer, tuberous sclerosis, cardiac hypertrophy, obesity. And what all these conditions have in common is they're due to inappropriate or ectopic growth, too much growth. You know, a tumor is a situation where cells are growing where they're not supposed to grow. More recently, TOR has been involved in another set of disorders, the so-called metabolic disorders, such as diabetes. In this case, it's not mutations which hyperactivate TOR, but reduce TOR activity, which also occur in humans in disease state. So, how do we know, for example, that TOR can cause cancer? Well, here's one experiment I will show you that we did in the lab many years ago. We made a mouse in which we could genetically hyperactivate TOR, and we do this in the liver, specifically in the liver. So we have a mouse which has hyperactivated TOR only in the liver, and the hyperactivation occurs at birth. So we have this mouse where we have hyperactivated TOR in the liver starting at birth. And what we find is these mice, we have to sacrifice them at 20 weeks. And we look at the liver, it has a very heavy tumor burden. So we've produced a liver tumor, liver cancer, hepatocellular carcinoma in this mouse due solely to the activation of TOR. There's a direct correlation between activating TOR and getting cancer. And we can, of course, do a further experiment to show that these tumors are due to hyperactivated TOR, because we can treat rapamycin, which, remember, binds TOR, and I didn't tell you it also inhibits it. And when we do that, this mouse is completely fine. The liver is completely normal, despite the fact that it is the same mouse that developed these tumors when they weren't treated with the rapamycin. So this tells us that rapamycin can, of course, be used to treat cancer, and that is why it's become now a famous drug in the treatment of cancer. Now, there's yet another, I would say, dark side to TOR. And here we have to think about what TOR is doing in the adult when we're no longer growing. TOR is still active, but now instead of driving cell growth, it takes on a new role. It takes on driving the aging process. These cells start to age, and of course, since the cell is the unit of life, it's also the unit of aging. If our cells age, we as an organism age. And that has indeed been shown that TOR promotes the aging process. It hasn't been shown in humans, like you can see on this slide here, because it's much more difficult to do experiments like this in humans, but it's been shown in yeast, flies, worms, and in mice that you can inhibit the aging process. You can extend the health span of these organisms by inhibiting TOR. And there are many ways of inhibiting TOR. You can either treat either the mouse or whatever organism you're using to do the experiment with rapamycin. You can also restrict the diet. Remember, TOR is activated by nutrients, so if you give less nutrients, TOR is less active. Or for experimental models, you can actually do genetic inactivation as well. And this has all been done with many different organisms, of yeast all the way to mammals, and it's been shown that indeed, if you inhibit TOR, you extend the lifespan. So targeting TOR is not only a strategy for treating disease, it can also be a strategy for preventing the aging process. So I hope you appreciate that this was a rather fundamentally important set of discoveries. And TOR has been celebrated ever since. We discovered it in 1991. It's now grown to a huge field on its own, producing these 7,000 papers per year. And the community, at least used to when it was not so overwhelmingly large, it's now thousands, is to get together to celebrate the landmark events in the lifetime of TOR. For example, here's a party we had to celebrate the 20th anniversary of TOR in 2011. I, likewise, have been fortunate to be celebrated as well as the face of TOR, I suppose. I've had a chance to go travel the world, where I've received a large number of awards. As Professor Terracciano told you, this has allowed me to meet the giants of industry, kings, presidents, people, I have to say, as a researcher, never dreamt in my wildest dreams I would ever meet. I love going to the lab. I don't seek out these kind of interactions, but it came with the discoveries. Here, for example, is in San Francisco with Larry Page, the founder of Google. Here's with Senator Nakasone in Japan. Here, the best company of all, my family, my wife and my daughter. Paris, here, the king of Sweden in Stockholm. But perhaps the most pleasant of all was at the Quirinale with Mattarella last year. I had the pleasure of spending the afternoon at the Quirinale where I met your president. It was an extremely enjoyable time for me and my family. It's an event I will not forget. So So here, I have to say, although I've received the prizes, I didn't do the work. I certainly have to acknowledge all the students, postdocs, and collaborators who work in the lab. I've had over 120 of them in the years working on TOR. They come through the lab and move on. And here's just a very small subset of these young, bright, and eager researchers. And they come from all over the world. For example, U.S., India, Switzerland, UK, France, Switzerland, Germany, Canada, Italy, U.S., Israel, Japan, and the list goes on. It's been a real privilege to work with all these young people over the years. And this is also a theme you heard about this morning, how you need to promote bright young people. And this is certainly confirms, I think, that notion. So the story is not finished. We've generated important new knowledge in the area of biomedicine. And there's also a theme you heard this morning, the importance of generating knowledge and transforming it. You saw that in the video that was shown shortly before I came on stage. So we've generated this knowledge, and we feel very happy about this, but we are still not satisfied. There's still a missing step here. And that step is, of course, to apply this knowledge to improve the human condition. And this is, again, a theme you heard over and over again this morning. So how can we do this? How can we take this next step? Next step is, of course, to start a company. And this is what we've done. We started a company in Basel, a biotech company, with a focus on developing therapies not only for cancer, but also for non-cancer indications. And we're developing small molecules which inhibit this TOR pathway, these TOR signaling molecules, and the ones around them which mediate cell growth. We have had some success here too. For example, this molecule here, which we call bimiralicib, it is an mTOR inhibitor, a TOR inhibitor. mTOR is often the term used for mammalian TOR. And we recently did a phase 2 clinical trial with this molecule to treat a skin condition, a precancerous skin condition called actinic keratosis. And the response rate was 90%, and it was with little to no toxicity. So we think this molecule is now, we're planning phase 3 trials, and we think it has a wonderful future ahead of it. So, and hopefully we'll be able to apply then our knowledge about TOR, which led to the generation of this molecule, which we made in collaboration with a chemistry student at the University of Basel. Okay, I'd like to return now to the title of my talk, The Personal Story of Discovery. I think you can appreciate the discovery we made. But why is, and I also said, this story reflects the importance of fundamental research in biomedicine, fundamental curiosity-driven research. And why is fundamental research important? I'd like to say that although I'm promoting fundamental basic research here, it does not mean I do not believe in translational research. I do. In fact, we've translated our basic findings. So translational research is also extremely important, I would say essential, but you need to build a foundation of fundamental research to then translate. You have to translate something, and what you translate is the basic finding. You translate that to the clinic. So in these days, in this day and age, there's a rush to translational research, which is good, but don't forget the fundamental research. You need to build the translational on something. So why is fundamental research important? Of course, it allows discovery. You can find the unexpected. I think our work reflects that. We never expected to discover TOR. We never expected to discover that cell growth is in fact controlled, and there's this very elaborate mechanism doing it. And making discovery is not easy because by definition it's unknown. You cannot control when you make a discovery. So you have to be willing to do this fundamental curiosity-driven research where you don't know where it's going to take you. You go and you do an experiment, you get a result, and try to understand it, and then if you interpret it the right way, you might find something completely unexpected. But your mind has to be prepared to meet this. Okay. So you need to do basic curiosity-driven research. This, of course, leads to innovation, for example, new therapies. I think I provide an example of that as well. This then positively impacts society, and you heard from your president the importance of what goes on at Humanitas in impacting society. This leads to greater health and prosperity. And even further down the road, this creates economic value, a return on investment. This is a return on investment not only by the agencies who gave us the money to do the research, but now hopefully there'll be a return on investment of the investors who are supporting this startup company, for example, we made. There's a return on the investment at every level, and I would argue that it's a very low-risk investment. I think it always provides a return. Okay, that was the importance of basic research. Now, the third part I promised you was how can we ensure success in fundamental research? I hope you agree that the story was a story of success. So how can we replicate this success perhaps in other places and other times? Is there a formula for success? And what lessons can we take from the story I just told you? As you know, there are no guarantees for success. But there are two factors which I have found can increase the chances. And those are the proper ecosystem, and the second is talent, having the right talent. And I'm very pleased to hear in the talks this morning that both these things were touched on, and how hard you're trying to build this proper ecosystem here in Milano. So again, I can confirm that it is the right way to go, and I congratulate you for actually having this vision. So let me talk about the proper ecosystem. This is a system which encourages this curiosity-driven basic research. That's how you're going to make the new discoveries, which can then be translated and lead to benefit to society. Of course, this was also brought up earlier, you need adequate funding. And as I said before, I think this funding is a risk-free investment. It pays off. It pays off. And you also need very good, diverse colleagues. Bring people from all different walks of life, different countries, that don't necessarily think like you. They can think differently. You can exchange ideas and discover new things. I myself was fortunate to work in such an ecosystem. That ecosystem was Switzerland. Switzerland is a very small, landlocked country with natural resources. Yet it's a successful country. And you have to ask yourself, why? It really makes no sense. And the reason it's been a successful country is because some visionary, I've not been able to pinpoint who that person was, decided to invest in the brain in Switzerland. They really had no choice because we have no natural resources. And this is now paid off. So I encourage politicians in Italy to invest in the brain. I know you're trying to do that, but keep going. It's very, very important. Finally, the talent. What do I consider the important characteristics of talent? What are the people that have done all this wonderful work in the lab? What properties do they have, which I've noticed? And again, I was very pleased to see all these issues come up also in the talks. So what I have to tell you, again, is not a secret. There's no secret formula here. It's obvious, but I'm confirming it, letting you know, if nothing else, that you're on the right track. You have to have ambition. And here I would say that preferably in the sciences, it has to be moral ambition, the ambition to want to help other people, not necessarily ambition to have fortune or fame, but to help other people. This is what's important. If you have moral ambition, science is the right medicine is also the right career for you. It's obviously not the right career for everybody. I never went into science with the idea of becoming famous or wealthy because I've generated a lot of knowledge, and that's the wealth that I feel I generated. I'm very satisfied with that. You also have to be very bold. You have to challenge authority and facts. I've noticed there are a lot of military people in the audience, and so I'd like to point out that challenging authority is something in science, not the military. Of course, authority is very important in the military. But in science, you have to challenge the facts. You have to challenge authorities, the people who come with ideas. You have to challenge them on the validity of those ideas. And are the facts true? This is how knowledge develops. This is how knowledge progresses, by challenging all ideas and replacing them with new ideas. We would still be stuck in the dark ages if we didn't challenge ideas. And this is what we have to do in science. Curiosity, this was also brought up earlier today. You have to appreciate the importance of knowledge. Knowledge has to be important to you. Perseverance, you cannot be discouraged. In every career there's adversity. I know in science there's immense diversity, because when you do experiments you usually fail. You have to have passion. This also came up. You have to have the passion where you don't feel enough hours in the day for you. You can't pack enough into a single day of everything you want to do. You rush to the lab in the morning and you have to do your experiment, and you can't wait. You have to go home eventually, and then you have to come back the next day. You can't wait. This is the passion you need. I say you need it all in science, but whatever career you choose, I hope there is passion. Here I can quote Maxim Gorky: "When work is a pleasure, life is a joy. When work is a duty, life is slavery." And I believe this is true. And finally, of course, you need to be lucky. I think we were extremely lucky in making the discovery we made. We could also have been unlucky, and I would never have ended up on this stage talking to you. But you have to be lucky. But I've also noticed in the people who've come to the lab and been the lucky ones, those are the ones who prepared themselves the best. Those are the ones that worked the hardest. Those are the ones that read the literature. They prepared their minds so that chance favored them. They understood what they saw, and you can almost, to a certain extent, make luck. Not always, but you can understand discovery when you make it. And now, to close, I would like to all the faculty, students, and staff, Humanitas University, best wishes and much success in the academic year 2025-2026. Thank you.